Welcome to this next episode in our Emmes at Home series, a program developed by Emmes to share thought leadership safely and securely from their homes during a pandemic. We are pleased to welcome Supriya Menezes, who will be moderating a panel discussion on the challenges of managing rare disease studies in ophthalmology.
And I'd like to wrap up with asking each panelist to share a success story about your involvement in a rare ophthalmic disease study, or what inspires you to continue to participate in research around rare ophthalmic diseases.
Some of my experiences, some of my successes, the coloboma protocol that we've been doing and engaging social media as a lesson, albinism, creating new outcome variables and engaging the FDA, and Stargardt, again, engaging the FDA and using each patient as his or her own control based upon carefully obtained natural history study. And I want to thank Dr. Clemens and all those that have been involved in the MacTel 2 project, because the success of one group of people in a rare disease often can pay forward. So in this particular example, the fact that MacTel 2 showed that there was a relationship between retinal structure using an imaging modality, OCT, and retinal function. In the end, the FDA cares about function. And so the fact that that correlation exists now enables me, as somebody working in Stargardt, to say, you know, my primary outcome variable is going to be a structural outcome, because look at that result from the MacTel 2 phase 2 trial. So I think that there is a synergy often created with successes in rare disease clinical trials research. In terms of why I stay with it, one, I just find it very rewarding and very fun. And as Dr. Clemens pointed out, a lot of these conditions, if not nearly all of them, have no treatments. There's nothing that we as ophthalmologists have to offer patients. And so the fact that you can think of potential new treatments, try those, and hopefully do some good, to me, that's a really strong motivator.
I'd like to start with or discuss an example of a disease where we're still in the early stages of describing this condition and understanding it. It's in the natural history stage, and that is reticular pseudodrusen, which is a mouthful, but it's an interesting story. It's a condition that was described decades ago. It's very subtle, drusen-like changes. The Kleins here at the University of Wisconsin were one of the first to describe it, seen in fundus photographs. It really wasn't until OCT and even high-resolution OCT that we're able to better identify these pseudodrusen. We don't really know what they are. They're deposits under the retina, not under the RPE. And it really takes multi-modal imaging to identify. So we do know that with an autofluorescence, with OCT and infrared, we will get the best chance of identifying these new drusen-like structures. And they're important because they've been shown, they correlate with some worse outcome in terms of geographic atrophy. So these are older patients. AMD is not a rare disease. We thought this feature was pretty rare, but it turns out it's not so rare. So we're getting more information on how often we even see these atypical pseudodrusen. And now there is a natural history study underway. It is taking a lot of images. It does mean a fairly long visit for the patient, but with the help of Emmes, we are, I think, able to get really good information on patients with drusen, with and without those pseudodrusen, and try to understand what happens, what is the natural history. We have come up with a common language. I think that took a while. We've also come up with some imaging strategies. So it's evolving, but it is an example of what we certainly thought was rare. Turns out not to be so rare, but it's been difficult to find. So we're really working on kind of uncovering the mystery behind these pseudodrusen.
As Dr. Blody mentioned, MacTel, I was also a part of the MacTel and getting the structural outcome that is currently being used in the phase 3 study, I think for me was very rewarding. The phase 1 study for the neuroprotectant that is currently under study started when we were just understanding the natural history. So we had an underlying natural history study that was ongoing. And at that time, we had about 6 or 7 years of follow-up on those patients. And what we were learning was that visual acuity was slowly progressing in those patients. Actually, we had conducted an analysis, and on average, those patients lost about 1 letter of visual acuity per year. So, as was noted, the outcome for the FDA is usually best corrected visual acuity, and it's usually 15 letters is the bar. So, if we were to conduct a study, as you can imagine, in MacTel using best corrected visual acuity, it would be a very long, daunting study. This outcome in the phase 1, which was just a safety outcome, and that was okay with the FDA. We also used it as in phase 2, again, to collect more information and again, stronger information with regards to the relationship of structure and function, and was able to provide that information to the FDA and also to the European regulators, EMA, to show that, yes, indeed, even what we showed from the natural history data, which was a strong correlation with structure and function, was even stronger with our clinical trial phase 2 information, relationship with structure and function. From that information, they did allow for our phase 3 study, which was the pivotal study. So for me, that was very rewarding, especially given that MacTel is a disease where we have no treatment.
We appreciate your time today. For our viewers, if you have any additional thoughts or questions, we encourage you to reach out to Emmes. You may contact me directly or others in the Ophthalmic Therapeutic and Research Unit shown here. We also hope you will join us for our other Emmes at Home events. Thank you for attending and have a lovely day.